Welcome to the course Animal Diversity 1 Biology of Non-Chordates. Myself, Dr. Sneha, Assistant Professor, Department of Zoology, St. Joseph's College for Women, Vishakhapatnam. As a part of this topic from Unit 1, we are discussing reproduction in Protozoa. As a part of this topic, reproduction in Protozoa, we are going to talk about various types of asexual reproduction modes in Protozoa, various types of sexual reproduction methods in Protozoa, and later we are going to compare both asexual and sexual reproduction methods in Protozoa. After studying this topic, you will be identify different modes of reproduction in Protozoa, that is both asexual mode and as well as sexual mode. You can able to describe asexual methods such as binary fission and multiple fission, how they're going to occur, etc. You can able to explain the various types of sexual reproduction methods, what actually happens in phylum Protozoa, that is like conjugation and syngamy with examples also. You can able to analyze the significance of reproduction in creating genetic variation and as well as which helps them for further survival of the protozoa. So, protozoans exhibit both asexual mode of reproduction and as well as sexual mode of reproduction. During favorable conditions, the protozoans, most of the protozoans, they are going to reproduce with the help of asexual mode. On the other hand, whenever the unfavorable conditions like absent lacking of food, too much extreme heat, overcrowding, so under that type of conditions which comes under category of unfavorable conditions, so under that circumstances the protozoans prefer to go through the sexual type of reproduction. Okay, so by in this way the protozoans are going to ensure rapid multiplication basically with the help of asexual mode. On the other hand, even during unfavorable conditions, so they as they're going to choose the sexual type of reproduction, so they can exhibit genetic variation. So this is how the asexual and sexual mode, both modes, the protozoans are going to reproduce. So first we're starting with asexual reproduction in protozoa. Asexual reproduction is most common mode occurred under favorable conditions as we already discussed. So for going through this asexual mode, only single parent is actually involved as a part of this process, where the main characters of this type of asexual mode, if you're going to see, there is no fusion of the gametes because only single parent is actually involved in this process. And this asexual mode of reproduction ensures rapid multiply of the protozoans, and as well as because of this, as only single parent is actually involved in this process, so the ultimately the results, the daughter cells, whatever they're going to produce, are genetically identical. They are, we can call them as like clones actually. So in protozoans, we can able to observe, as in the slide also you can see that, so all these are various types of asexual methods what we can observe in the phylum Protozoa, like very commonly seen is binary fission, next is multiple fission, third budding, plasmatomy, encystment. So all these are parts of the type of asexual modes which are exhibited by various protozoans. So let's start with binary fission, which is most common method exhibited by most of the protozoans. So where one single parent, the mature parent cell divides into two equal daughter cells, which actually involves first of all the mitosis, that is what division of the nucleus, which is followed by later the cytokinesis process, that is what division of the cytoplasm. So again, further, this binary fission is actually divided into three various types depending upon the plane. That means the fission, it is whether it is occurring longitudinally, transversely, like basing on that, the binary fission is actually three types. The first one is simple or irregular type of binary fission, where this type of fission doesn't include any exact plane. The best example for this is amoeba. followed by the second type is longitudinal binary fission. As the name itself is indicating, the fission, the axis is basically longitudinal. So the example for this category is euglena. Followed by the third one is transverse binary fission, where the axis is across the transfer section. The best example for this is paramecium. So these are the various types of binary fission. So let's talk about these in detail one after one. We are starting with simple binary fission, right? So as I already said, that simple or irregular binary fission is a type of binary fission or a sexual mode, basically, where two daughter cells are formed, but there is no exact plane. So this is it is basically temporary type of the plane of fission is actually decided, right? So because of this, it might happens irregularly. So there is no fixed axis is actually present, right? So it is most commonly seen in amoeba, right? Here in this diagram, you can able to clearly observe that how it is going on, as amoeba is doesn't have a constant shape, irregular shape, right? So now if you're going to see, whenever the amoeba wants to go through this type of binary fission, which is known as irregular, right? So now I clearly see here, first of all, the amoeba feeds actively. It increases its size, the single parent. And later, we can clearly see the axis is almost all started here. And before going through this axis, even you can clearly see the division of the nucleus is also going on here with the help of mitosis. And ultimately, once that division of nucleus is properly done, you can clearly see this single parent is ultimately divided into two daughter cells. So this is how the irregular type of binary fission we can clearly able to see in amoeba. Next, followed by the second type of binary fission, that is what longitudinal binary fission, where the axis of the fission is basically longitudinal, that is from anterior to posterior end, right? So where the parent cell splits into two equal daughter cells, and each resembles with first one exactly resembles with the second one, or left one resembles with the right one. Actually, they are exactly like a carbon copies, what we can say. So this longitudinal binary fission includes lots of steps. So let's talk about those steps, starting with the growth phase. So once the best example for this is euglena. So we're taking that, considering example as euglena. So whatever euglena parent, it wants to go through this longitudinal binary fission. First of all, it feeds well, increase its size. So that is what part of growth phase. Once this growth phase is over, then it starts with the next step, that is what duplication of organelles, such as like flagellum, or it might be chloroplast, whatever organelles are there. So they're going to start getting duplicated. So each daughter cell is going to get the same type of the cell organelles. And once this is also done, then it enters into the next step, that is what karyokinesis or nuclear division. As I already said, the nucleus is going to divide with the help of the mitosis process, where the two identical daughter cells are going to receive the two nuclei. Once the karyokinesis is over, then even we can clearly see, so see in this diagram you can clearly able to observe the same thing, right? So here this is how the nucleus is going to divide, and in the meanwhile the karyokinesis is going on. We can able to clearly see here, also see that at the anterior end, even the axis, the fission is actually already started, right? So once the karyokinesis is fully over, so this fission, that is what furrow which is formed, it is going to deepen further, right, till the posterior end, right? And once they are going to detach from each other. So this is how basically exactly the two daughter cells are going to be formed as a part of longitudinal binary fission. Next, after this, followed by the third type of binary fission, that is what transverse binary fission, right? So, which is very commonly seen in ciliates, and the result, as a result of this transverse binary fission, also the two daughter cells are going to be formed, and but here. The daughter cells are going to be formed anterior and as well as the posterior. The anterior daughter cell is actually known as spotter and posterior daughter cell is known as opisthe. Again, let's talk about the steps involved in this growth phase. The transverse binary fission. Again, it starts with growth phase where organism is going to uh, keep on eating and feeds actively and increase its size. So once that growth phase is over, then it is going to start with nuclear division. Okay. So now if we're going to say in as I told you now that this transverse type of binary fission mostly occurs in ciliates. So all of you know that ciliates are going to have two different types of nucleus. That is what they want to exhibit dimorphic type of condition. That is what one is macronucleus and another one is basically micronucleus. Okay, so now as a part of this transverse binary fission, the both macronucleus and as well as micronucleus, they're going to divide. So now let's talk about that. So first we will start with macronucleus. Macronucleus divides by amitosis process, okay, but on the other hand the micronucleus divides by mitosis process. Okay, with the help of these the karyokinesis is going to be over. Then it starts with, in the meanwhile, once the karyokinesis is also happening, side by side the duplication of organelles is also going to be happen, such as like oral grooves, cilia, contractile vacuoles, and development of new oral aperture. All these things are going to happen side by side. And in the meanwhile, from external side of the paramecium's body, if you're going to be observed, and we can able to observe a development of a constriction, like you can clearly see in this diagram here, right? Okay, see, constriction, one furrow is basically appearing here, right, which is going to be that means deepens. First of all, it starts from the sides of the paramecium body, later it actually deepens further, and because of this ultimately the cytokinesis is also going to be over. So because of this result, the two daughter cells are going to be formed, right, which is one of them, it is known as spotter, and second one is known as opisthe, right? So this is how even transverse binary fission is actually happens in paramecium or most of the ciliates. So this is about the different types of binary fission which occurs in protozoans. So after binary fission, the multiple fission, where this is also one type of the asexual type of mode of reproduction which occurs in protozoans. Okay, so as we just now talked about binary fission, bi means what? Because of this type of fission, only two daughter cells are basically forming. Okay, but here as we are saying is multiple fission, right? So that means, see, multiple daughter cells are going to be formed with the help of same process. So that is what, first of all, it starts with karyokinesis process, and which is followed by cytokinesis process. But here what is the difference is, because of this fission, this type of fission, the multiple number of daughter cells are going to be formed. So because of this, it is named itself as multiple fission. Okay, the same thing you can clearly see in this diagram also in this slide. Okay, where first of all the nucleus is going to divide repeatedly with the help of mitosis process continuously, where it is going to form enough number of the like hundreds of nuclei are going to be formed once this the karyokinesis is over. So each nucleus is going to receive a little amount of cytoplasm as a part of cytokinesis, as you could clearly see the same thing. So once it is also over, all the nuclei are going to move towards the peripheral side of the parent cell, which ultimately is going to be separated from each other. Okay, so this is how at a time many daughter cells, like hundreds of daughter cells, can be formed with the help of one multiple fission, right? So after the fission, then we're talking about one more type of asexual mode, that is what, budding. So the same thing you can clearly see in the diagram also. So from the parent cell itself, one small outgrowth, like you clearly see here, which is known as actually bud, right? Which is actually originates from the parent body. Later onwards, it is going to grow in size and ultimately detaches from the parent cell, like here, see, right? Which is ultimately grow into a new new individual. Okay, so this is how budding actually happens. The best example, lots of protozoans actually are going to exhibit mostly the sessile type of protozoans. Sessile means where they can't move for their entire lifetime, right? So mostly in sessile type of protozoans like Verticella also, which can't move, right? So in that type of protozoans, we can able to clearly see this type of asexual mode. That is what budding. That means apart from Noctiluca, Verticella, all these type of protozoans, they're going to reproduce by budding. So after this budding, one more type of asexual mode, that is what plasmotomy, right? So here, as a part of this plasmotomy, if the protozoan is exhibiting multinucleate condition, right? Multinucleate means the parent cell is having like more than two number of nuclei, right? So then in that case, that is known as multinucleate condition, okay? So in that type of protozoans, like Opalina is the example for this, right? Where the parent cell is already having exhibiting the multinucleate condition, where ultimately the division, the division of nucleus is going to be happen, right? And as a part of this plasmatomy, it is not see, in the binary fission we have talked about, after mitosis one nucleus, it is going to be received by one daughter cell and another nucleus is going to be goes to another daughter cell, right? So but in this plasmatomy, as the parent itself is indicating or having the condition of multinucleate, so the both daughter cells are going to be formed. They are not going to get equal number of nuclei. Some daughter cells might get less number of nuclei. Some might get little more number of nuclei. Okay, so that is about the characteristic of plasmotomy. So that is what, see, each daughter cell gets some nuclei. Okay, so this is also one type of asexual mode what is actually seen in a few protozoans. Moving to last category of asexual method, that is what encystment. Encystment is not exactly a type of asexual mode, but it helps the protozoans to withstand to the unfavorable conditions, as you could clearly see here. So this protozoan is experiencing unfavorable conditions like lacking of food, or it might be experiencing extreme heat. So during that condition, what this protozoan is going to do is it is going to secrete a thick layer, a cyst like layer around this entire protozoan body, right? And sometimes it was observed by scientists is inside this cyst, they might reproduce. That is what multiple fission might happen. Okay, because of this cyst formation, it helps in what survival plus as well as dispersal very easily from one place to another place. So this is also one more type of mode of asexual production what we can able to observe in some protozoans. So this is about various modes of asexual type of reproduction what actually happens. So we have discussed about binary fission, multiple fission, and even we have talked about plasmotomy, budding, encystment. Okay, so now we are moving to the next type of the mode of reproduction, that is where it is known as sexual reproduction, where the main characters of the sexual reproduction, if you're going to see, right, it involves the fusion of the gametes and as well as exchange of the genetic material, right? So under what circumstances protozoans go through this sexual method, as we already discussed in the first, that is what, during unfavorable conditions, and it is actually required for the protozoans to rejuvenate their genetic variations basically, and as well as because of this, the protozoans can increase their adaptability to new environments. So that is what the main significance of the sexual reproduction. So let's see how the protozoans are going to, by what methods they're going to reproduce sexually. Okay, so this slide indicates shows the various types of sexual methods what we can able to observe in protozoans. So among all these four, the first two one are very important and very commonly seen in ciliates and flagellates mostly. That is what conjugation and syngamy. So these are very important the modes of sexual reproduction which is followed by autogamy and endomixis. So let's talk about these method. So first we are starting with syngamy. Okay, so syngamy is a type of sexual reproduction which actually involves the fusion of the two gametes. The two gametes are actually formed where the fusion of the gametes is going to be happen. So in that, whenever the gametes are basically forming, it might, in some protozoans, one gamete might act as male gamete, one might act as female gamete, and ultimately because of fusion of these gametes the zygote is going to be formed. Okay, so these are the various types of syngamy we can observe in the protozoans, which is further divided into three categories. That is what isogamy, anisogamy, and oogamy. Okay, so now let's talk about what are these. So isogamy, as the name indicating, iso means some protozoans, they're going to produce the gametes which are similar gametes. Similar gametes means it might be male or female. Both of them are going to exhibit same size, same shape. Okay, so that type of gametes are known as actually isogametes, and the process is known as isogamy. And because of the fusion of the gametes, and ultimately the zygote is going to be formed, which is going to further grown, and ultimately the young protozoan is going to be formed. Okay, so in most of the primitive protozoans, this isogamy type of sexual reproduction actually happens. Okay, so now moving to anisogamy, which is completely opposite to the isogamy. Isogamy, where we are saying iso means what? Similar gametes, right? So but in the anisogamy, what actually happens means, aniso means the dissimilar gametes. That means one gamete might be very larger in size and shape comparing to the other one, which is much more smaller. Okay, if the fusion occurs between this type of dissimilar gametes, then that type of syngamy is known as actually an isogamy. Okay, now oogamy, which is most commonly seen in parasitic protozoans, where properly egg and as well as sperm, that means a female gamete is clear cutly formed, which is obviously bigger in size, which is non-motile of course, but on the other hand the male gamete basically, right, which is much more smaller in size, which is very active, which can verily move, right? So that is what. So that is why this type of syngamy is basically known as what? Oogamy, which is very commonly seen in our very well-known studied protozoan parasite, that is what? Plasmodium. Okay, so this is how syngamy actually occurs in protozoans. Then followed by the second type of sexual the reproduction method, that is what? Conjugation. Okay, so in this conjugation the protozoans are not going to form any gametes, but the protozoan itself, for the temporary time only, the protozoan is itself acts as a so as a part of conjugation, it is a union between the two protozoas. The mature individuals, for the time being, they're going to basically temporarily unite, right? And they're going to exchange the genetic material, and as well as because of this exchange of genetic material, so they can create genetic variations for the next generation. So that is how conjugation actually happens, right? Because of conjugation, see, as I'm saying, it does not increase any number. Okay, but why it is part of sexual reproduction? Basically, see, because of this conjugation, what is the result is genetic recombination, because of exchanging of the genetic material between two mature protozoan individuals, that actually happens. They're going to simply exchange the genetic material. So that is how conjugation is basically very important. Okay, so now let's go through that the steps which involves as a part of conjugation. So first of all, it starts with pair formation. Mostly it occurs in ciliates. So we are studying the conjugation, what actually happens with reference to paramecium, right? So the two parameciums, first of all, they're going to come together side by side, right? And towards their oral surfaces, basically where the oral aperture is actually present. So towards that area, they're going to combine with each other, temporary union. So that's what I'm saying, right? So a clear cut, a small cytoplasmic bridge after that union, a cytoplasmic bridge is actually forms between those two parameciums, right? So once this union is done, right, so then what actually happens is all of you know that paramecium contains two nucleus, that is what, macronucleus, which is larger in size, on the other hand micronucleus, which is smaller in size, right? So macronucleus is not going to play any role in this conjugation process. So that's why once the temporary union was actually done properly, then the macronucleus which is present in these two individuals which are united with each other, so it is going to be disintegrate in both individuals. Okay, now once this conjugation is over, later it is going to be reformed again later, but okay, but here in the conjugation process the main role is performed by the micronucleus. Okay, so once that macronucleus is disintegrated, then the diploid micronucleus, it goes through the meiosis process. As all of you know that once the meiosis 1, meiosis 2 was done, so what is the product? The result is basically the four haploid nuclei are going to be formed. Okay, so among these four haploid nuclei, uh, remember the three haploid nuclei are going to be degenerated completely. Only one haploid nuclei in each individual of paramecium is going to stay, which is basically functional. Okay, one remain functional, the one haploid nuclei is going to remain, right? So now here what actually happens, this one haploid nuclei which stays, right? So here among this, if you're going to see, so this haploid nucleus again divide mitotically. Because of this mitosis, it forms basically what? Two pronucleus. The first nucleus, which is smaller in size, which is known as migratory pronucleus. On the other hand, which is little bit larger in size, that is what? Stationary. It is can't move basically. It is a non-motile category. Okay, now. So now once these two pronuclei are formed, the same thing I will show you in the diagram here. See, so now if you are going to see here in this diagram, like once this pronuclei are formed, see here, right, in both the temporary union was occurred here in between these two parameciums, right? So this is the bigger one is known as basically stationary pronucleus, and as well as the smaller one is actually known as actually the it can move, it is a motile pronucleus basically. Okay, now so once this two migratory pronucleus, so once they both are formed basically, the exchange is going to be happen. Exchange is going to happen means how? As the migratory pronucleus of this paramecium, it will migrate from here to here. Okay. On the other hand, the migratory pronucleus of this paramecium, it will move from here to here. Okay. So after this, the movement of migratory pronucleus, it is going to basically fuse with what? Pro, the stationary pronucleus. Okay, so the fusion of the both stationary and migratory nucleus is going to happen, and it forms what actually? Syncaryon. Syncaryon is going to be formed. So now this is what the end of the conjugation. So once the conjugation is end, the already united both parameciums, they're going to detach from each other, and they're going to later develop their macronucleus, as I already explained in the previous slide. So this is how conjugation actually occurs in paramecium. So after conjugation, let's talk about autogamy. Autogamy is something like a self-fertilization, which is also seen most commonly in ciliates like paramecium, okay, where in the same one single parent, it doesn't involve any two parents or two gametes, okay, now, where it involves basically see single individual actually, okay, single individual, where we can able to see like self fertilization, how in plants it happens. So this is also some kind of self fertilization where the nucleus in the same organism is going to fertilize with each other. As a resultant, there is no genetic variation. Okay, now, but this is also type of the reproduction is can be seen in paramecium. Okay, Coming to next is endomixis. So endomixis is also something like a, not like syngamy and conjugation. It is not that much important, but here also if you're going to clearly see here, so endomixis is basically, see, where we can able to observe the nuclear reorganization. Okay, now nuclear reorganization and fusion within one individual only. Okay, now so there is no like presence of two individuals as a part of autogamy, or it might be endomixis clearly. So this is also type of method which actually occurs in ciliates. Okay, but if you're going to compare all the four different types of sexual modes, so most commonly talked about is only syngamy and conjugation. Okay, so now let's compare both asexual and as well as sexual reproduction, what actually happens in the phylum Protozoa. As all of you know that in protozoans, especially in asexual reproduction, it involves only one single parent. On the other hand, in sexual reproduction, like it involves two parents, right? uh, okay, the two gametes basically, or it might be for genetic exchange. Okay, and the time period, like for an example, for asexual reproduction, it is very fast actually, the fission, like hardly in 2 to 3 hours the process is going to be over, right? So but on the other hand, sexual reproduction is little bit slow comparing to the asexual. Okay, but coming to variations, so with the help of asexual reproduction, as only one single parent is involved in this process, so there is no genetic variation. But on the other hand, in sexual reproduction, because involvement of the two gametes, involvement of the two different individuals, right, definitely there is genetic variation. So as all of you know that during favorable conditions, protozoans go through that asexual method. On the other hand, during often unfavorable conditions, the protozoans go through sexual reproduction. Okay, the example for the asexual is binary fission, which is most commonly occur, and example for sexual reproduction is actually conjugation. So total, if you, the summary of this entire reproduction in protozoa. So as all of you know that protozoans reproduces both by asexual and sexual method. Asexual mainly occurs through binary fission, multiple fission, etc. On the other hand, sexual reproduction mostly occurs as a part of conjugation and as well as the syngamy, which actually ensures the genetic variations. Okay, so reproduction in protozoans ensures its continuity, adaptability, and survival of the protozoan species. So this is the reference what is cited for this topic. Thank you for listening the class.